Thanks, Tony. I don't think that was impromptu. I think you'd been planning that. So thank you. So now we'll hear from Mark Richardson. Now, Mark is well known to the audience. Mark's based at King's College in London and has been prominent in this field for a long time and a good friend and good collaborator. So Mark will be speaking around goals and hot topics in epilepsy, mechanisms, model, prediction and control. Thanks, Mark.
Thank you very much, Mark. So good morning, everybody. I'd like to thank Levin and Mark and David and the organising committee for this very flattering invitation to kick off this session. So as Mark said, I'm a neurologist specialising in epilepsy, and I have a research interest, amongst other things, in exploring how the properties of large-scale brain networks allow seizures to occur. When Levin asked me to take on this session, I was initially very daunted because it's a very big topic. This is kind of life, the universe, and everything of epilepsy. So I've had to narrow things down and be highly selective for this brief session this morning. So if there are things that I don't mention, your favourite study, your own study, please just forgive me. I've just been very selective, and you might think that I could have chosen better examples, but they're the ones that I thought were interesting. So what I'm going to try to do is just give you a personal perspective on what I think might be goals and hot topics over the near future. And I'm going to articulate these goals and hot topics as a set of broad challenges. So I'm just going to lay these out in front of you, first of all. So the first challenge that I want to mention, and I'll elaborate on these in a minute, is to look at seizure prediction as a problem of signals processing, data mining, pattern recognition. We know, broadly speaking, as a method that this works, but how far can we get with it? Then a second challenge is just to reflect on the fact that actually we know plenty about epilepsy, and are there things that we can do in a hypothesis-driven sense, using knowledge from experimental models to try to identify variables that we might track and that might predict seizure onset. And then we heard some interesting contributions yesterday about the role of modelling in this context. So a third challenge is to consider how we might use multi-scale models to inform the development of seizure prediction methods. And in particular, can we use models as a kind of magnifying glass or a way of simplifying multidimensional data that we monitor so that we might track particular time-varying parameters and see that those are heading towards seizure onset. And then a fourth challenge is to think about moving beyond EEG. One of the drawbacks of EEG is that it's difficult to measure, and it's certainly difficult to measure from the scalp for long periods of time, and it's highly invasive to measure it intracranially for long periods of time. So there are other things that change, other physiological variables that change around seizures, and some of them are very easy to measure. So should we be looking at some of those things? And then the next challenge is just to reflect on the fact that seizure prediction is not just an intellectual exercise for some fun, but should be something that has a purpose. So how do we then close the loop and find a treatment that could respond to a predictive signal? And then finally, something that Steve Schiff talked about yesterday is about how we can measure and how we can treat and control. Where do we do those things? It's very tempting to think that we know where we might need to measure and treat most relevantly, but I think that we probably don't know that. So let's start with this first challenge. So since we're in Melbourne, I'm yet another person mentioning the Cook et al. Study. And this was a study that I'm sure you all know well. And just to remind you that this was a study that in some highly selected patients, so these are a series of individual patients, and this is time prior to seizure onset at which a warning was put into the system, so a time at which the seizure was forecast. And these red lines showed a median prediction time for each of these subjects. And these are hundreds of minutes, so these are somewhere around 1 or 2 hours in most cases. And that looks great. Looking at the table just draws attention to a number of things. So first of all, the probability that seizures could be forecast, these p-values are very low for most of the subjects. So this seems to work. But if you look at the percentage of time spent under a high seizure warning, it averages about 25% of the subject's time was spent under high seizure warning. And about a third of seizures were not forecast. So it works, but it has some limitations. A study that was published a year later, so this is last year, from the Epilepsy Eye Consortium, again, this was mentioned yesterday, looked at a number of different methods, and this is a complicated study with a lot of take-home messages, but I'm just going to focus on one thing. So this diagram here shows the minimum prediction sensitivity against the false positive prediction rate, and this is the number of subjects. So essentially what this shows is that there are some subjects who can achieve a very high prediction sensitivity at a very low false prediction rate. But you can't probably read the numbers here, but these are just handfuls of subjects out of 200 plus that achieve that really fantastic prediction capability. But there's lots of patients here who at lower sensitivity and higher false prediction rates also have predictability of their seizures. So a question, and this is a very naive question, is how might we move these patients from this low sensitivity and high false prediction rate into this area? How can we improve the performance of the methods? So the challenge here might be how can we refine signals processing and pattern recognition algorithms to be faster, more accurate, easier to develop, applicable to more subjects? And in addition, something that I haven't mentioned, how could we uncover more information, perhaps through using a stimulus, probing stimulus, rather than just passively observing the system? So the next challenge is in a contrast to the previous. Rather than data mining in a relatively hypothesis-free manner, can we make use of what we know about seizures to identify specific phenomena which occur reliably prior to seizures? So what can we learn from experimental models? So this study is from an experimental animal model, and I'm not going to go into the details, but just to show you this figure. So this is the inter-seizure interval, and this is the number of seizures that had this particular time since the previous seizure. And you can see that this is very clearly bimodal. So there are seizures that occur at short inter-seizure intervals and seizures that occur at long inter-seizure intervals. This is just a long-winded way of saying that seizures tend to cluster. So the occurrence of seizures is not a random thing. There may be some underlying patterns that drive seizure occurrence. And this second example shows that there may be some particular brain states in which seizures are much more likely to occur. So this might be part of why seizures appear to be not a random phenomenon. So these are some observations from human nocturnal frontal lobe epilepsy, and this just shows a number of particular sleep states, and again, I'm not going to go into the details, but here, different sleep states are contrasted with each other, and the number of seizures occurring in one state versus the other are compared. And what can be seen here is that these nocturnal frontal lobe seizures occur in very specific sleep states and not in others. So there are clearly some seizure predisposing brain states. So at these relatively long timescales, seizures don't occur randomly. At much shorter timescales, there are phenomena that appear to anticipate the electrographic onset of seizures. So this is a study looking at hemodynamics in association with the seizure focus. So again, I'm going to skip a lot of the details, but this is a focal epilepsy model in which, amongst other things, arteriolar diameter in the vicinity of the seizure onset zone was being continuously measured. And I'll just take you down to this part of the figure. So this shows constriction of the arteriole just prior to electrographic seizure onset. So this is some seconds prior to seizure onset, relatively short time interval. But this appears to occur, so this is the constriction of the arteriole prior to the electrographic seizure onset. And we know that there are also electrographic phenomena that can occur in this pre-ictal period. So in this study, and again this was mentioned yesterday, there are some preictal large spikes that occur prior to seizure onset. Again, it's within the seconds prior to seizure onset. So it does look as though, in these particular examples, that there are some observable phenomena that occur prior to seizure onset. So therefore, can we make use of these specific observations in experimental studies to identify brain states in which seizures are more likely to occur? And can we identify observable phenomena that reliably change prior to seizures? So in a hypothesis-driven way, try to detect states in which seizures are more likely to occur. We actually know rather a lot about the basic mechanisms of epilepsy and seizures, but the data that we obtain from patients, of course, is a very incomplete account of the brain system that generates seizures. So we heard yesterday some ideas about using models to tell us more about the underlying processes. So could we construct a model that generates our incomplete observational data based on what we know of the system? And then could we attempt to use the model to interpret the data, perhaps revealing that the system is heading towards a seizure onset? And let me give you some examples of what I mean by that. So I'm showing this as an example of how a model might reveal that transitions in a system are driven by relatively few underlying parameters. And I hope Steve, who's in the audience, won't mind me presenting this. And I'm going to make some naive comments about this, Steve, so please don't give me too hard a time about this. So this is a model of neuronal membrane dynamics. And this phase space diagram in the middle here is bounded by two parameters. So there's the extracellular oxygen concentration, the extracellular potassium concentration, and as those two things vary, the dynamics of the system varies in some very characteristic ways. So there are some regions of this space, for example, this region, in which the dynamics generated are those of seizures. There's another large region of this space where the dynamics generated are spreading depression, and so on. So rather than using the model to generate different kinds of dynamic activity, it might be possible to take some data and invert the model and try to infer the underlying parameters. And to my naive way of thinking about this, it might be possible to find that there are particular parameter combinations where there is a trajectory as the parameter moves towards a particular region of that space where a seizure is more likely to occur. And just to show that that's not wild fantasy, this is an example that shows that, at least to my mind, this might be feasible. So again, I'm not going to go into the details of this, but this is a model of the generation of seizures in the hippocampal system. And then these are three parameters of the model, and then these are the values that the parameters take in different states of the system. So this is the interictal, the preictal state, sorry, I can't even read this. Yeah, the seizure onset and the ictal state. And you can see in this preictal state that the combination of parameter values is different from the interictal state. Now, this study just took chunks of data and then averaged the parameter values over that chunk. But it might be possible to take small pieces of data and track how these parameters change over time. So this is not a study of seizure onsets. This is a study of how the parameters in a system that generate spike wave discharges vary with each cycle of the spike wave discharge. So again, these are parameters of the system, and rather than just taking large chunks of particular states, the parameters are tracked with each cycle of the spike wave discharge, and so show particular trajectories that are reproducible between subjects. And in this diagram here, the parameters follow a particular trajectory across the phase space. So it might be possible to find that a parameter is heading in a particular direction towards where a seizure might be going to start. So there are a lot of challenges in pursuing this kind of line of investigation, including developing useful models, identifying suitable parameters which change prior to seizures, having methods of model inversion that can be run in real time and on short data segments, and exploring in particular whether this is any better than the kind of relatively hypothesis-free pattern recognition approach that one might take in the same data. So much of what I've said so far has been about EEG, but I've also mentioned hemodynamic signals and some other things. But we know that seizures are not just a phenomenon that can be observed in the brain, but there are signals in other parts of the body that might be observable and might be easier than EEG. So there is some evidence that cardiac autonomic function changes prior to seizures. So this is a study from a colleague at King's College London a few years ago now, where amongst other things they tracked something related to cardiac parasympathetic drive, which in the normal state showed these characteristic oscillations, and then prior to seizures those oscillations were lost. It's much easier to collect signals from the heart than it is from the brain. And then this is the study that we saw before, where there was some hemodynamic phenomena that changed prior to seizures, and we saw here that seizures associate with particular sleep states. And okay, EEG was used to determine these sleep states, but we know that, for example, very sensitive accelerometry might give some broad ideas about sleep states. And again, that may be easier to measure in the long term than EEG. And some of you may be wearing one of these. So there's been an explosion in consumer wearable devices that track various activity-related or various physiological parameters. A lot of people with epilepsy associate a number of things happening prior to seizures with subsequent seizure onset. And in many instances, those things are not proven or even have been disproven. But, you know, as this kind of thing becomes really ubiquitous and many people are wearing them, including our epilepsy patients, I really don't know what this may reveal, but I'm sure that this is something that's worth exploring further. So an important challenge is whether to explore this area, and if so, how far and what kinds of data to collect, and whether we need to be collecting large-scale, long-term monitoring data, as with EEG. So this whole area has no purpose unless we have some kind of beneficial treatment that can respond to a seizure predictive signal. And I really don't think I have a clear idea what that treatment would be. I assume it's going to be based on one of these kinds of technologies, either an electrical stimulation device or perhaps an intracerebral drug delivery device or perhaps something else. I'm also not sure whether I should be expecting a treatment based on a seizure prediction signal to be passive until some kind of signal crosses some kind of threshold and then it just gives a zap to the brain and hey presto, everything's restored back to normal, or whether this would be a continuous phenomenon that controls the brain state, preventing seizures from starting. And then finally, another substantial challenge is about where to measure to detect a seizure predictive signal, and then where to treat. And this is really a very complex area that I'm not going to go into in any detail. But just to reflect on that, at least from theoretical work, it does seem likely that there may be more than one mechanism driving the emergence of seizure-like synchrony. It might be driven by a focal node, or it might be driven by a network. And even if we think that a seizure onset is driven by a focal node, it's very tempting to think, well, we should measure from that node and we should treat there. But all I would say in response to that is that the model of resective surgery for focal onset epilepsy assumes that we can find where the seizure is driven from, remove that piece of brain, and the patient is then cured. But we know that that's often not the case. And although in some instances those focal onset epilepsies can be well treated by epilepsy surgery, We know that there are many people who fail that surgery, and we also know that many of the important treatment-resistant epilepsies probably don't have that kind of focal onset, but may have an onset in a distributed network, in which case certainly I don't have any clear idea about how one might go about measuring from that network or treating the entirety of that network. So in summary, these are the challenges that I can see are important for the near future. In many ways, I hope you disagree, because that would set up a very interesting discussion over the next half hour or so. So thank you very much for listening.